welcome in the hall. We also have a live stream, so I hope somebody or some people are watching outside this hall. We have 60 seats nowadays in this hall, and maybe tomorrow we have to bring it back to 30. So it's very exciting times. We're taking our measures, we're keeping our distance even in this hall, so I hope you feel comfortable sitting here. For us on this side, it's a bit difficult looking at all the blue little cards on the chairs saying, don't sit here, don't sit here, don't sit here, don't sit here. But we do see you, so very welcome at this lecture of Studium Generale. When I met Burgert Blom, the speaker of tonight, or actually he mailed me and he said, I'm doing such interesting research and I'm getting so far that I really want to share it with people. I was, of course, very interested because he gave a lecture in our PAS Festival, I think 2 years ago, very interested. And the PAS Festival is for us a stage where young researchers can share their research with an audience. But also, you know, as a start, we do want to ask them at the end to come back for our program in this hall or at the university. And his research is really interesting. I don't understand it at all, I think, although he tried to explain me and I thought, oh yes, yes, I think I understand. So that's a lot. But I'm not a chemist, so, and then I thought, well, this is interesting research. It is needed to share this. And he is such an enthusiastic researcher, not only in what he researches, but also in what he does and how he does it. So tonight it's going to be on the development of novel metal-based anti-cancer agents. I don't know if you understand what that exactly is, but he's going to talk about it, of course, in detail. And the interesting thing is also that he does this research within his faculty with students. So students support him in a program of the FSE, is this? Maastricht Science Programme, Faculty of Science and Engineering, yes, where he works. And he also will tell you a little bit about how this research is done and then how these discoveries, interesting discoveries, are made together with students. Welcome, Burgert Blom. He will talk for about an hour and then we have time for some questions from the audience. Burgert Blom.
Thanks, Rob, for the introduction. And I'd like to thank everyone for coming tonight. As Rob pointed out, these are very difficult circumstances we're in. So it's nice to actually see some faces again. And this is the first time I've actually given a lecture to people, living beings, live in about half a year. So I feel like an academic again. Yes, about 2 years ago, I did give a talk at the PAS Festival here in Maastricht, and that was sort of at the beginning stages of this work, which I've been doing for approximately 4 years here. So I thought it would be a good idea to sort of expand on that. And so I'm glad to be here today to share some of these results. So you see some images here, and these are actually images from the work that we've been doing, these beautiful crystals. The compounds that we're working with contain metals, and they're active against different types of cancer. And so we're going to be looking at metal-based drugs today. So the crystals you see on the left and on the right are actually of some of the compounds we're going to look at. And in the center, you see actually what some of these materials look like. They have these orange and red colors. And that's because they contain transition metals. So I'm a synthetic chemist. I make molecules. That's what I've been doing for the last 20 years. And in particular, I work in a subfield called organometallic chemistry. And that's basically a field which lies at the interface of organic and classical organic chemistry, the chemistry of carbon, and inorganic chemistry. Okay, so what we're trying to do here in this field is make molecules that actually have some sort of a bonding between these elements in the middle. They're called transition metals, elements like iron, manganese, nickel, cobalt, rhodium, etc. And the elements on the right, you see with a second arrow there, the so-called p-block elements, in particular carbon, silicon, germanium, tin, and lead. So these compounds are called organometallic because you're combining something organic, carbon, with something
inorganic, a metal. And the compounds themselves tend to be actually quite reactive. So you see here, for example, depicted at the bottom is sort of a very general formula of one of these compounds, where E actually is one of the elements on the right, one of the P block elements, and M is a transition metal. Actually, also the purple elements at the bottom, the so-called lanthanides and actinides, also fall into this category. And so I've denoted this bond between M, which is a metal, and E, which is a P block element, with a dotted line. And the reason why I use a dotted line is because the bonding interaction between the metal and the main group elements is sometimes very interesting. And you can use that interesting bonding situation for a variety of different applications. For example, catalysis, right? So we're not just making these compounds because they're fundamentally interesting and important, and you learn something about how atoms bond together in a very fundamental way, but you can also use these molecules to actually do interesting things. And so you can use them also in medicine, and that's what we're going to be talking about tonight. So for the last 13 years or so, actually what I've really been focusing on is getting the transition metals in the middle and also these F block elements at the bottom, the lanthanides and the actinides, to bond to group 14 elements. Those are carbon, silicon, germanium, tin, and lead. And they form very interesting sorts of molecules that you can exploit for a number of different things. And so that's sort of what I focused my research on. And being a synthetic chemist, it's all about making the compound in a laboratory, isolating it, characterizing it spectroscopically to make sure that you have what you think you have, and also doing various structural investigations, for example, with X-ray analysis. So we're making beautiful compounds and we're studying them. We're studying their reactivity, and we're doing stuff with them, catalysis, for example. So these compounds are very interesting and fundamentally very important. We don't just make them, we also study them from a theoretical perspective. So we do something called typically density functional theory to really understand the nature of these molecules. And so once we've understood their reactivity, we can use them for certain applications. So again, for me, typically it's exploring the reactivity of the metals in the center with group 14 elements, carbon through lead. Now, this sort of chemistry is technically very demanding, and it's technically challenging because the compounds you make tend to be very reactive. And they're reactive because the metal to carbon bond is typically quite reactive. And so you have to do these reactions with very special equipment. And what you see here is actually a picture from our lab. This on the left here is a so-called Schlenk line, which is standard for this sort of chemistry. It enables you to maintain very reactive compounds under an atmosphere of nitrogen or argon, which is an inert gas. So very often if you expose these sorts of compounds to the atmosphere, they undergo decomposition, or they even burn on contact with air, so they're very reactive. The compounds we're going to be talking about this evening are actually organometallic, but they're not reactive, because we can use them actually as drugs, okay? So they're an exception to this. But typically in organometallic chemistry, the compounds are very reactive. You have all this very special glassware that you need to use. You see on the right here, these are called Schlenk flasks. You see there's a tap attached to it, which enables you to actually do manipulations of these compounds under an inert atmosphere to protect the compound. And there is other standard equipment like this. This is called a glove box. This enables you to manipulate compounds in a very pure atmosphere of nitrogen or argon. So less than 1 parts per million oxygen or moisture typically if the glove box is functioning correctly. And so it's a safe environment for these reactive compounds. And this is just sort of what it looks like in practice, again from our lab. This is actually a reaction that one of my students was doing. And you can see the vessel is attached with this rubber hose to the Schlenk line. So it's maintained under a positive pressure of an inert gas, because the compound in there in this instance was pretty reactive. So it's an experimental challenge to work with these compounds, and it's technically quite difficult. So for the last 13 years, this is really the summary of what I've been doing. I started off in my PhD looking at fundamental investigations, particularly looking at the nature of the bond, just fundamentally, between transition metals and group 14 elements, for example, silicon through lead. So that's interesting because those sorts of compounds hadn't been made, and we were interested just looking at the nature of the bonding interaction. And then I moved to Berlin, and I was doing something similar, except I focused on silicon, getting silicon to bond to a transition metal. And there I was looking at the use of these compounds in catalysis. What is catalysis? It's using some sort of an entity that enables a low energy pathway for a reaction to take place. And we made some interesting discoveries there. And when I came to Maastricht, I decided to change gears and move in a medicinal direction, but using these compounds and the knowledge from my past, and actually trying to see if these compounds were biochemically active. So I jumped into this field, but actually the chemistry really does have some background in my previous career in Berlin and in Bonn. So that's kind of what gave me these ideas. So that's what we're going to be talking about tonight. Medicinal applications of these specific organometallic compounds, in particular combining group 14 elements with transition metals. So the problem is cancer. This is a very big problem in the world. It's one of the leading causes of death. Millions of people succumb to this disease annually. And what you see on this cartoon here is really a very simple representation of what cancer is. It's really the uncontrolled replication of unhealthy cells. Cells that in an uncontrollable fashion, as you can see on the right there, undergo division. And this results then in the formation of tumors, invasive tumors. And in normal cell division, it's a very controlled process you see on the left here. And it is also controlled and regulated by various proteins. So the moment the cell becomes damaged, the replication will discontinue. So this is a cardinal difference between normal and cancer cell division. The one is controlled and the one is completely uncontrolled. And this is a major problem. And the incidence of cancer is increasing annually. So this is certainly a problem worth our attention scientifically. Now, if we look at some examples, these are scanning electron micrographic images of different types of cancer. We have there some examples: lung cancer, breast cancer cells. You get different types of lung and breast cancer. This is, for example, triple negative breast cancer. That's an electron micrographic image of one of the cells. And lymphoma at the bottom, you can see the red blood corpuscles sort of squeezed in between these cancer cells. And there's a sort of an electron microscopic image of a brain cancer cell. You can just see by looking at these cells that something is very wrong with them morphologically. And so the cartoon I just presented you showing this replication is actually quite an accurate depiction of what these cells end up looking like. And there's this uncontrolled division that then results in the formation of tumors. Okay? And that's where the problems begin. So traditionally, there's really three approaches to dealing with this disease, certainly in the last 50 years. Surgical removal of invasive tumors, if they're localized, is usually the first line of attack. And then that's typically combined with chemotherapy and radiotherapy. What we're going to be talking about today is chemotherapy, which is the use of chemical agents to actually kill the cells, induce apoptosis. That's the fancy way of talking about it. Stop cell proliferation. And so as a chemist, this is of course something that you can make a contribution in. So the story with chemotherapy is actually very interesting in terms of metal-based chemotherapy. And it starts in 1965. There was an American scientist named Rosenberg, and he actually serendipitously discovered the action of this compound here. This compound is called cisplatin. It's a platinum-based drug. You can see on the left the chemical formula. Chemists like to represent the structure of molecules in a three-dimensional way. And that's sort of what a chemist would use to represent this compound. On the right is a space-filling model. That's what it looks like, with each of those balls representing atoms. Anyway, what happened with Rosenberg serendipitously is he was actually investigating the effect of an electric field on E. coli. And what he found is that if you expose E. coli in a medium to an electric field generated by platinum electrodes, the cells actually increased to 300-fold size. This is something very strange he didn't expect. And what he actually discovered was actually that the platinum electrodes were undergoing corrosion in the medium he was using and generating this compound at the bottom here by accident. And this compound was responsible for this different behavior in this E. coli microorganisms. And so that's essentially how they discovered this compound. And after many tests, about 13 years later, it was FDA approved in the United States. That's a long time ago, that's 42 years ago, but this compound is still being employed in anti-cancer therapy, even 42 years later. The problem with these platinum-based agents, called platins, of which this is the first generation, is that they have many side effects. And actually, anyone that has known someone that has cancer who's been treated with a platinum-based drug, typically in combination with other non-metal-based chemotherapeutic agents, knows what these side effects look like: hair loss, vomiting, alopecia, just to name a few. And so, although there's been further generations of platinum-based drugs more recently, carboplatin, which is also currently used, oxaliplatin and heptaplatin as well, these are slightly better than cisplatin in terms of their side effect profiles, but still not great. So we can certainly do better than that. And so this is where the idea of actually doing metal-based chemistry for new anti-cancer agents actually comes in. And cisplatin, discovered in 1965 and FDA approved in 1978, has been extremely well studied, the mechanism of how it actually induces cell death. And this video from a colleague of mine at the University of Rhode Island, used with permission, shows us how cisplatin actually induces cell death. So that's the formula
Cisplatin is
of cisplatin. You can see it's nicely square planar. And it's sort of drifting around in the cytoplasm. And it's somehow going to interact with DNA and do something to it, and we'll see in a moment what it does. So there it is, making its way to the cell, and what it will encounter when it gets to the cell are these proteins on the surface which enable the drug to be taken up either actively or passively. So it diffuses into the cell across this membrane, active or passive. There you see it going in by both of these pathways. And finally, it goes into the cytoplasm and makes its way to the nucleus, where the DNA is residing. There you see a DNA strand. Now what happens now is critical. The two chlorine atoms, which are green here, will basically undergo a process called equation and be replaced with water molecules. And then it's going to bind to the DNA. And in particular, it binds to two consecutive nitrogen atoms on the guanine bases of the DNA strand. So guanine is one of the nitrogenous bases in DNA. It contains nitrogen, and it can bind to this platinum center. And when that happens, you have a pretty stable adduct. We call this an adduct, and this induces bending in the DNA. It bends the DNA molecule. So it damages the DNA. And in the process, since the DNA, as you can see there, is bent, a protein comes along to try and repair the damage, a DNA repair protein, and this protein gets stuck. And it gets stuck for something quite complicated. It's called a pi-pi stacking interaction of these atoms over here. But the point is it gets stuck, and the whole thing just freezes. And so it prevents cell replication, because you need DNA for a cell to replicate. So the cell then eventually dies. So this is the mechanism of cisplatin, very well understood and very well investigated. But as I've just said, these platinum drugs have many side effects. So what about other transition metals? Well, there are many of them. We just saw that periodic table. And in particular, for about the last 20, 25 years, scientists have been looking at the element ruthenium, which is kind of a close neighbor of platinum. It's 2 groups away. And in particular, there's been one compound that's been synthesized about 20 years ago, 18 years ago. That's the chemical structure by a colleague of mine in Switzerland, with whom I actually currently collaborate, Paul Dyson. And that compound is called Raptor-C, Raptor-C. And it's been shown that this compound is very effective in combination with other drugs in vivo in actually killing cancer cells. So this is kind of a starting point for my research, is focusing on ruthenium. And in particular, what I wanted to do was look at the synthesis of new compounds that we can actually test to see their efficacy with respect to different types of cancer. The question is, in the overall grander scheme of things, where does my research fall? Well, it would fall somewhere here, kind of in the early stages, early drug discovery and screening, somewhere between there and preclinical studies. So there's still a long way to go before the stuff that we've been working with could potentially be used as an actual drug clinically, but this is really how this research goes. So it's sort of blue sky work with the potential of pre- and post-clinical studies. And so what we're looking at now are perhaps some mechanistic insights, synthesis and drug screening on different cell lines. My strategy is to make bimetallic systems. Now, this is not an entirely new strategy, but the combination of metals that I've chosen is new. And you can imagine if you want to get 2 metals to come together to form a bond, there's a number of ways that you can do it. You can just put the 2 metals together and get them to form a chemical bond, as I've depicted here. Those metals will be decorated with other substituents, but this is one possibility. Another possibility that we can imagine is that we have 2 metals in close proximity to each other, which are kind of bound together by what we call a tether, a rope, a kind of a chemical rope. And we can vary the length of this rope, in principle, and see what effect that might have on the anti-cancer activity of this bimetallic compound. A third possibility is that you actually make some sort of a salt, kind of like sodium chloride, that you use to season your food, except instead of having sodium and chlorine, you have some sort of a transition metal which is positively charged and a negatively charged counterpart. And we've looked at all 3 of these strategies, and I'm going to show you sort of what progress we've made. Now, why would we want to make a bimetallic system in the first place? Well, it's kind of, as I say at the bottom there, if you have 2 metals, you could imagine a situation where you have some sort of a synergic effect, where the 2 metals work together and in the end perhaps have a better cytotoxic or anti-cancer activity than just the individual metals on their own. So the whole point of why we are doing this is hoping that we have some sort of a synergic interaction. So that's the rationale behind it. And so the starting point is this ruthenium complex from my colleague Paul Dyson. And these compounds, in contrast to most organometallic compounds, are exceptionally stable. And this is a very nice playground for a chemist, because you can actually do all sorts of interesting chemistry to these compounds. You can, for example, imagine manipulating this carbon ring that sits kind of like a hat on the ruthenium center. You can play all sorts of games there and decorate it with different substituents and change its properties. You could, for example, imagine playing around with this phosphine over here. That's just one of the groups attached to the metal. It's called a phosphine. You could imagine abstracting or changing the halide, and you can also, for example, consider making it ionic. So there's a lot of games you can play with these molecules, and if you change the structure, it's going to have a direct effect on the biological function. That's the point. And the point is to try and design molecules rationally that are actually going to do what you want them to do. Another thing that you can do, which is the first step in what we did, was actually to replace one of these groups with another metal. And so the starting point for me was tin. So one of those strategies I just showed you is putting two metals together in close proximity, and it's quite easy to make these sorts of compounds. So that's where we started. We started making compounds containing tin atoms. Sn is the chemical symbol for tin, the element tin, it's a metal, in close proximity or directly bonded to the ruthenium atom. And so what you see depicted on the left there is a chemical reaction scheme for how one actually synthesizes these compounds, okay? Chemists like to write down chemical reaction equations which represent the molecules that they're making. And so this is a chemical reaction scheme where actually it's quite a simple reaction in this instance. You start with this ruthenium compound on the left, that's a representation of its three-dimensional structure, and you react it with tin dichloride. And what actually happens is the tin dichloride, SnCl2, inserts itself into the metal halogen bond, and you can see there what we get as a product is ruthenium bound to SnCl3, tin trichloride. Now, you can play all sorts of other games, which we did, which is to decorate it then with different substituents. For example, you can replace the phosphine there, P is the chemical symbol for phosphorus, with other substituents. And that's important for biological structure-activity relations. So one of the most exciting methods that one can use is something called X-ray crystallography. So this picture that you see here is actually an experimentally determined structure, three-dimensional structure of one of these compounds that we synthesized. So essentially you grow crystals of this compound, expose it to X-ray radiation, and from the interaction of the X-rays with the electrons in the atoms of the molecule, you can position them in three-dimensional space to an exceptionally high degree of accuracy. So you will see more of these structures as we go on. This is kind of unambiguous proof that you've made what you think you've made. So this is just one example of what the three-dimensional structure of one of these prodrugs looks like. The other game that we played was actually to make an ionic system. So here you see the compounds on the left are neutral, they're not charged, but the compounds on the right, well, the one compound on the right is actually charged. It's got a plus and a minus. Why did we do that? Well, if it's ionic, it probably is going to have a higher solubility in biological media because it's like salt, and salt dissolves in water. That was our thinking. So we tested these compounds on 2 cell lines, but before I get to the results, it's important to know what you want in a drug. You want a drug that's going to selectively go for cancer and not for healthy cells. It doesn't help if you have something that just kills everything. That's useless. You want a high selectivity index for the cancer cells. So that's the one thing you require. And the other thing you require is actually a high activity with a relatively small amount of the compound. How do we actually determine these things? Well, we do in vitro testing. This is the first stepping stone to actually determine the activity of the compounds. So this is not being injected into a living organism. It's being done in a petri dish, basically. But you have a cell line, and then you subject, basically, at different concentrations of your drug that you're testing, the anti-cancer activity of, you subject it to cells and you incubate them. And effectively, after a certain amount of time, let's say 48 hours, you actually count the number of surviving cells. And if you plot that, actually, you get a curve that looks like this, a sort of a sigmoidal shape, where essentially we quote a so-called IC50 value, which stands for half maximal inhibitory concentration, and that's a reflection of how potent this compound is with respect to inducing cell death, okay? And so what you would typically do is you would test this on a cancer cell line, and you would test this also then on healthy cells, and then you can get the selectivity index to see if it's preferentially going for the cancer cells or not. So the lower the IC50 value, the more potent is the agent, right? So we want low numbers, because low numbers means the compound is very active biologically. So if we look at these compounds, this is sort of the first study, we see something very interesting. We have here 2 different cancer cell lines. These are from my collaborator in Switzerland, Paul Dyson, and co-workers, these results. And so we can see we've tested it here on ovarian cancer cell line, 82780, and cisplatin-resistant ovarian cancer, 82780 cis-R, and then we tested on the healthy cells, the kidney cells. Now what you immediately notice, if you look at this series of compounds, is the neutral compounds bearing this tin C1 through C3 are pretty inactive, right? They've got very high IC50 values. But if you look at compound C4, which contains the tin atom in the salt, its activity is higher than the analog that does not contain tin. So the conclusion is, making the compound ionic, it seems to have something to do with solubility. You can enhance the cytotoxic activity of the drug if you make the thing more soluble. So there seems to be an issue with solubility here for C1, C2, and C3. But what you notice if you look at the data of C4, essentially cisplatin is still the winner on ovarian cancer with a value of 1.1, so the lower the number, the better. And effectively, you see in terms of selectivity, C4, although it performs better on 82780 cis-R, which is the cisplatin-resistant cancer cell line, it doesn't have a particularly good selectivity, okay? So nevertheless, for a first stab, interesting results. And so what we learned from this initial study is that solubility seems to be an important parameter. So you need this balance. And if the compounds are too insoluble, which the neutral compounds were, we have very low cytotoxicity. So the next study that we performed was to try and make a series of neutral compounds, so not ionic, but neutral ones, that exhibited higher solubility in aqueous medium. And how can you do that? Well, chemists have all these tools at their disposal. If you, for example, imagine a molecule that has a tail attached to it with an OH group at the end, that OH group can interact with water, and that should make the thing soluble. So the trick here is simply to make the same molecule but attach a tail to it such that that tail can actually interact with water, and that can enhance the solubility, and perhaps that improves the cytotoxicity. And so we proceeded to do that, and here you have sort of an overview of the chemical reaction scheme. This is quite complicated, but effectively this is just the way we synthesize these molecules. At the bottom, 2, 3, 4, 5, 6, and 7, those are the products. That's just the way of synthesis in the lab, and effectively you can see that we also have an X-ray structure of one of these compounds where you can clearly see you have this tail attached to this hat that's sitting on the metal, and you have this trichlorostanol group, which is what we call it, an SNCl3 group, bound to the ruthenium center. So the question is, if we attach this tail, does it improve the cytotoxicity? And well, first of all, it does certainly improve the solubility, as you can see here. So that's actually just a sample of this compound dissolved in normal water. And you can see it has a beautiful orange color. And so that means it's partially soluble. So the trick of attaching this tail has apparently worked. And on the right, you just see the colors of some of these compounds. And you see the difference between having no tin atom present and incorporating a tin atom, SN, into the bond of ruthenium. It makes these beautiful differences in color. Okay, you have beautiful colors for these compounds. So these compounds were tested. This time, unfortunately, we didn't get the IC50 values, but we did get a sort of a qualitative idea of the relative cytotoxicity of the compounds. This work was done from another collaborator at the University of Cape Town in South Africa, Sharon Prince, and essentially, and a colleague of mine at the chemistry department, Siyabonga Ngubane, with who I also collaborate, and his PhD student. And we tested this on a breast cancer cell line. And this specific breast cancer cell line is called triple-negative breast cancer. And you can see across the board, the compounds, we have slightly enhanced the cytotoxicity of these neutral compounds compared to the first set of results that we got. But essentially, cisplatin is still, you can see by the low cell survival rate at those two concentrations still clearly outperforming all the other compounds. So we've made an incremental improvements by improving the solubility, but it is still not outperforming cisplatin. And so that's a pity. So from these two studies, we've learned that solubility plays an important role. Incorporation of tin can sometimes lead to an enhancement of cytotoxicity, but it seems to be very strictly dependent on the relative solubility of the compound. And so the question is, what about if we go from tin to germanium? Now, germanium, if you look at the periodic table, is kind of the little brother of tin. All right, it's in the same group as carbon. And so we did all this chemistry with germanium with a student, Conor Deacon Price, who's now at the University of Amsterdam. He did a beautiful thesis with me, and he made all these compounds. Again, we have these complicated looking chemical reaction schemes. It's just how we synthesize these compounds. And we basically did what we did with tin with germanium. And so what you get out of all of this is essentially these 5 compounds he synthesized. You see, again, we've played those chemical games. We've varied the nature of the substituent. We've changed, the ring that's sitting on the metal. Sometimes it has this aliphatic group attached to it, which increases the water solubility. Sometimes it doesn't. We varied the phosphine. But we made this homologous series of germanium compounds. And I should mention that the reason why we did the tin and the germanium chemistry in the first place is not just because I'm a tin and a germanium chemist. It's because if you actually incorporate the tin and the germanium onto the metal, you tune its reactivity in such a way that it can actually bind faster or better to a specific biochemical target, okay? So it kind of primes the prodrug to actually do its binding to DNA or perhaps to some protein involved in cell death better. And what happens with germanium actually is it does that very well. And if you take these compounds and you dissolve them in water, actually they immediately undergo a so-called ligand exchange reaction, which means that water immediately binds to the ruthenium center. And that actually, surprisingly, initially I thought was a good thing, because that means the compound should be very active biologically. But if you actually do the tests, again, these are results from my colleague in Switzerland. Here we just did it on A2780, ovarian cancer, and HEK293, the healthy cells. You see a complete reduction in the cytotoxic activity despite high solubility. And the question is, why is that? Well, the attachments of the trichlorogermyl group, that's what we call this GeCl3, the germanium, actually resulted in compounds that are so extremely unstable in a water medium that we think the compounds undergo rapid hydrolysis, that's the process, induced by the presence of this germanium group to form the compound you see on the right there, where one of the chlorine atoms has been replaced with a water molecule. Of course, there's lots of water in biological media. And this process is required to happen at some point before the molecule binds to a specific chemical target to induce cell death. But if it happens too quickly, then essentially it can potentially bind to a biochemical target that's benign and that doesn't have any influence on the cell death. So we think that this process happens too rapidly, and we see that spectroscopically. We have evidence for that, and that effectively explains why we have such low activities biologically for these compounds, for ruthenium. But it's quite interesting, because if you now change the metal, and these are recent results that are unpublished, and you go from ruthenium to osmium. So if we look again now at the periodic table, ruthenium and osmium are also sort of brothers. Ruthenium is the little brother and osmium is the big brother. You see there in group 8. So if you do the same chemistry with osmium, which we've done, then actually the picture is completely different. And what we have here, these are results from another collaborator at Cotton University in India. You actually see that if you change the metal from ruthenium to osmium, you arrest this instability in solution, this aquatic instability, and the compounds again exhibit a fair amount of cytotoxicity. And what you see here are two different cell lines, human lymphoma and a type of carcinoma. And these are actually apoptosis studies compared to cisplatin, the control, where you can see again cisplatin does a slightly better job because red means that the cells are dying or undergoing apoptosis, but orange means that there's some activity. So you see for this specific compound at different concentrations, 1, 5, and 10, you have some a significant amount of cytotoxic action, which is not the case with ruthenium. And that makes chemical sense because osmium, the big brother of ruthenium, actually is more stable with respect to this ligand exchange process. These are actually just some results from these osmium compounds that we've made on these two different cell lines compared to cisplatin. And you can clearly see actually that cisplatin actually outperforms the compounds still, but actually our germanium compounds actually don't do too badly. So we've learned something else. If you change the transition metal from ruthenium to osmium, then you get a secondary effect. So there are lots of parameters we need to investigate. What we've also actually, yes, this is essentially the take-home message here. This process of, let's say, hydration or equation is much slower in the case of osmium than it is in the case of ruthenium. That means the compound is stable for longer in a biological medium. It has time to get to the cell, then undergo the process, and then do its job. And that's consistent with the chemistry of osmium versus ruthenium. Ruthenium is a lot more reactive than osmium. So this is actually a rare example of an osmium compound that is more reactive biologically than ruthenium. There are very few examples of those. So that's kind of interesting. And recently we've actually started getting our teeth stuck into the mechanistic understanding of how these drugs actually work. And what we think is actually happening based on these in silico studies, this is from my collaborator, Dr. Verma, we actually think that these drugs actually target anti-apoptotic proteins. There are three important ones. These are proteins that actually prevent cells from undergoing cell death. And it seems, based on our docking studies, that these drugs actually combine and form adducts with these three proteins, BCL2, BCL-XL, and MCL1. Those are the three proteins, and you can actually see here at the bottom how these drugs, in fact this drug is one of these osmium compounds, actually interacts with these three proteins, blocks them, and that then in effect causes cell death, because these proteins are responsible for preventing cell death. So if the drug binds to it, it can actually shut down that process and the cell can undergo apoptosis. And you see at the bottom there, this is quite complicated, but you can actually see how the drug interacts with those different amino acid residues in certain pockets on the actual proteins. So this is the mechanism we think that's responsible for this class of compounds, but these are just, let's say, in silico studies, computer simulations, and so actual biological data is still something that we need to accumulate and do more detailed mechanistic studies on this new class of compounds. So we've looked now at combining tin and germanium with metals. Sometimes we get interesting effects. In the case of osmium, we enhance the effect. In the case of ruthenium, it depends on solubility. So there's all sorts of complicated second-order factors going on. But another thing that we can actually do is, coming back to our scheme of making bimetallic systems, is we can install a chemical rope between, we can install, well, I'll get to the chemical rope in a minute. We can make ionic systems. So instead of having a neutral compound, we have a salt. And that's precisely what we did. This was done by another student of mine, Nicolo. And he made these compounds. These are their chemical structures. Now, they're fundamentally different from the molecules we just saw. In the compounds we just saw, the germanium and the tin was bound to the transition metal. And here we have the germanium and the tin as a counter anion. Notice it's got a plus and a minus charge. They're not bound together. They're a salt. So the question is, what is the biological activity of these compounds? And this is very interesting. So this is from my collaboration at the University of Cape Town. That's just the X-ray structure of these compounds. You can see that the tin and the germanium atoms are very far apart from the ruthenium. So it really is a salt, okay, like sodium chloride. And it turns out that these compounds are exceptionally active and by a substantial margin actually outperform cisplatin, which is the standard positive control. And in particular, if we look at these two cell lines here, MCF7 is a model for triple-negative breast cancer, and progesterone and estrogen receptor-positive breast cancer, and MDA-MB231 is a cell line corresponding to triple-negative breast cancer. You actually see that you have a dramatic improvement in the cytotoxic activity, much lower than cisplatin on both cell lines for these different compounds. And you see the same thing for tin, so that's for germanium. And in the case of these, well, there's another example of germanium, really across the board, essentially dramatic improvements. So for the first time, we have agents that outperform cisplatin, which is really great. And we see that fundamentally these compounds are different from the compounds we saw moments ago because they're salts. So again, you have this notion of solubility potentially playing a role. If we look at the selectivity indices of these drugs, which is also important, by comparing the cytotoxic activity to a healthy cell line, in this case MCF12A, which is in fact breast epithelial cells, we see actually that we get very promising selectivity indices for all of these compounds. In fact, across the board, except for compound 5 and MCF7, we have values in excess of 2. And according to the literature, if you have a selectivity index greater than 2, that tends to be an agent that's promising for more clinical studies. So these are very promising agents, and not only are they very cytotoxic and have promising selectivity indices for breast cancer, but they're also stable long-term. So what we see depicted here actually is a so-called clonogenic assay where you can actually assess the long-term stability of the drug. This is over a period of 14 days, and we've essentially selected 2 of the drugs here that contain germanium, and we see a nice dose-dependent behavior of the drugs as a function of basically cell death. And so what you quite often have with drugs is they have a short-term capacity to induce cell death, and then the cancer starts growing again. But these compounds actually seem to, at least for a 2-week period that we did this analysis on, be stable and in a dose-dependent fashion suppress cell growth of the cancer cells. So that's very promising indeed. And so this is really exciting because we have compounds here that are quite active and fundamentally new. So the third strategy I want to just mention briefly this evening is actually this business of installing a chemical rope between 2 metal centers. In other words, a tether. We call that in chemistry. So we've looked at these 2 strategies, but what about this one? So what if we imagine a bimetallic system which is held together with some sort of a linker, where we can actually have a neutral molecule, or we can imagine even an ionic molecule, where the 2 metal centers are bound together. And so the idea that we had here was actually something quite interesting, I think, and it's something we're still exploring. And the idea here is actually, if one looks at cancer cells, typically, they have a very high concentration of so-called transferrin receptors. These are receptors that are responsible for the regulation of iron uptake in cells. And certainly cancer cells have a much higher density and number of these transferrin receptors than normal cells. Now, the idea we therefore had was actually, once if you imagine making a bimetallic compound where you have two metal centers, one being iron and the other being ruthenium, something like this, bound together. And the idea is it's a kind of a Trojan horse strategy where the iron end of the molecule, the iron terminus, should then go for the high density of the transferrin receptors, and then actually that can be used to sort of pull the molecule in through the membrane into the cell, and then the ruthenium side of the molecule can actually do its bit. So it's kind of like a sneaky way, potentially, to increase the concentration of the drug in a cancer cell, because after all, they've got a higher concentration and number of transferrin receptors, and so the iron center of the molecule is kind of used to chaperone in the other molecule. That's the strategy. And it really is a sort of a Trojan horse approach, because once the receptor has actually taken hold of that iron side of the molecule and pulled it into the cell, then the ruthenium can do its job and actually induce apoptosis. So we proceeded to make these compounds, and you can see here, this is again the chemical synthesis, but essentially there's a very neat and efficient way that you can couple iron with ruthenium. This is the chemical synthesis. It's essentially a one-step process. And so we made a series of these compounds, and you can see, if we look at these two molecules, they're identical in every respect, except the one just has a different substituent or group sitting on the ruthenium. Okay? We have to do that so that we can see if there's any effect with different substituents. But there you can see we made those two compounds, and there's this linker between the iron and the ruthenium, this rope, this chemical rope, which consists of 3 atoms, 2 phosphorus atoms and 1 carbon atom, so the 2 metal centers are actually quite close together. And for the sake of completeness, we also made a dinuclear ruthenium complex, so 2 ruthenium atoms, because we wanted to see if having iron present actually indeed does increase the relative biological activity of the compound. So if you replace that with ruthenium, you kind of have, let's say, a test case to see if there's a dramatic difference replacing iron with ruthenium. And indeed, if we look at well, that's the X-ray structure of one of these compounds. You can see it's a beautiful bimetallic complex with, the zigzag orientation of this chemical tether. Incidentally, again, this is from X-ray crystallography. And if we look now at the data, this is from my colleague Dyson in Switzerland and co-workers, indeed we see that in this series of compounds, these bimetallic compounds, 1 through 4, well, 1 through 3 actually, that the winner actually across the board is compound 2, which indeed is the heterobimetallic iron ruthenium complex. What's particularly interesting is that if you compare the numbers to compound 3, which is the bimetallic complex, with 2 ruthenium centers, you see a dramatic 15-fold decrease in biological activity on A2780, and also a decrease in activity substantially, in fact, across the board on all 3 lines. And so that is some sort of evidence that there is a sort of a transferrin-mediated uptake mechanism for these compounds. At least having iron presence in the molecule substantially enhances biological activity. So that's a very nice result too, and you can see that the numbers, again, outperforming cisplatin. And also the selectivity indices are not too bad. We have additional evidence that kind of indirectly supports this transferrin uptake mechanism coming from something called density functional theory, which is actually a way that you can, on a computer, calculate where the electrons are sitting in a molecule. So these balloons that you see here, they look like balloons, the green and the red balloons are actually a reflection of electron density in the molecules. And in particular, if you're a chemist, you're interested in something called a HOMO and a LUMO orbital. Those are kind of the orbitals, or that's where all the action is for chemistry. And if you look at these 2 molecules, you can see that these orbitals are strictly located on the iron terminus of the molecule, okay? And what that basically means is that any chemistry is going to be initiated at that end, not at the ruthenium end. So if the first step is actually some sort of binding to a biochemical target like transferrin, certainly based on these calculations, it's going to be the ion center that docks onto the transferrin. And so we have some indirect evidence based on the relative cytotoxicity, but also based on the density functional theory calculations that this is in fact the mechanism. However, we in the future plan to do detailed transferrin binding studies to confirm this. The next question is, what if we increase the length of the rope? So this was another student of mine who addressed this issue. And so we noticed in this first generation of iron ruthenium complexes, we had 4 atoms between the 2 metal centers. What if we just add an extra one in there? It's possible to synthesize these compounds. This very talented student of mine did it. That's what these compounds look like. Beautiful orange colors. Some of them are green. And not only did she look at increasing the length between the iron and the ruthenium center, but she also looked, that's the chemical synthesis of these compounds, it's quite straightforward. But she also looked actually at making di-iron complexes, right? So what is the effect of not having iron and ruthenium tethered together, but having iron and iron? So a dinuclear iron complex. And what you see here is actually a very surprisingly straightforward synthesis to one of these dinuclear systems. And actually there on the bottom left, you actually see an image of the structure of one of these compounds tethered with one of these long chemical ropes. You can also do all sorts of other things. You can, for example, play chemical games and replace what we had previously here, which is an iodine atom sitting there, and you can, for example, replace it with a CH3 group. That actually increases the lipophilicity, the fat solubility of the compound. And so if you subject these to cytotoxic tests, here we have the results on A2780 Cesar from my colleague in Switzerland, we see actually something very interesting. First of all, it would seem that compared to the previous study, increasing the size of or increasing the distance between the 2 metal atoms seems to lead to a slight decrease in the biological activity, a slight decrease, not substantially, but it's slightly lower, however substantially higher than cisplatin. And if we look at compounds 6 and 7, which are the di-iron complexes, we see a complete loss of cytotoxic activity at all. So that's another interesting result, because that tells us you need ruthenium in the molecule for it to be active. But what we also see is actually replacing an iodine atom here in compound 6 with a methyl group actually does result in some improvement in the cytotoxic activity, and that's because we've made the thing fat soluble. So what we learned from this study, and this is still ongoing, we are looking at expanding this study, we are looking at expanding this study comprehensively at this stage by making a full series of these compounds and actually trying to do structure-activity relations on it to really get our teeth stuck into exactly what length is optimal. But certainly we've uncovered a new interesting class of anti-cancer agents which seem to be quite active in vitro, and we seem to have, in terms of this iron ruthenium story, this is a very interesting development. So in summary, actually we've looked at 3 different strategies tonight. We've looked at combining ruthenium with a p-block element like germanium or tin. We saw that in some cases you have pretty promising biological activities, but sometimes you don't. We looked then at a second strategy, which was to make ruthenium complexes containing germanium and tin, but as salts, and those delivered excellent results, outperforming cisplatin substantially. And finally, we saw the difference between changing the metal from ruthenium to osmium. That also has an effect. And lastly, we looked at systems which are bimetallic, where we have a chemical rope between the 2 metal centers, in particular the strategy of attaching iron to it and taking advantage of the increased density of iron transferrin receptors on the cancer cells, which seems to certainly be a strategy that works, at least from our data so far. And the preliminary data that we have so far seems to suggest that indeed these compounds actually are being uptaken in the cell by sort of a transferrin-mediated pathway. So we are currently exploring all of these fronts further. We are looking at other combinations of metals. We're looking, for example, at rhodium and iridium as well. Those are other transition metals which exhibit interesting cytotoxic activities. We are continuing our work with osmium. And what we are going to really get our teeth stuck into in the future is really looking at the biological behavior of these systems and doing detailed mechanistic studies to try and understand rigorously how these new classes of agents are actually working, because it might be that their mechanisms are completely different to conventional drugs. And we saw that with osmium, initial mechanistic studies in silico seem to suggest a mechanism of deactivation by targeting anti-apoptotic proteins. So this might be something very interesting, and our future work hopefully will uncover new vistas in these new types of agents which are bimetallic. I would like to end off by, I'm going a bit over time here, but I'll be done in a few minutes, by thanking these people. These are my collaborators. I'm collaborating with a lot of people internationally. I think that's very important in science. And in particular, I would like to thank my colleague in Cape Town, Professor Sharon Prince and her team. We have a great collaboration going also with my colleague at the chemistry department at the University of Cape Town, Dr. Ngubani. And also my colleagues, you see the rest of the colleagues here, Professor Dyson, who's really, I'd say, one of the fathers of transition metal-based anti-cancer therapy based on ruthenium. We have a very nice collaboration. And all my X-ray work, those beautiful structures that you saw, are done by these two, Judith and Christoph, in Austria. We've known each other for many years, and they're always sending me these beautiful X-ray structures. And the calculations that I'm doing to understand the electronic structure of the compounds are being done in collaboration with my colleague here in Maastricht, Dr. Romano. And he always sends me those pictures with those nice balloons, but they're not balloons, they're actually a reflection of where the electrons are sitting in the molecule. And there's a few other people, for example, high-resolution mass spec, which is important in characterizing these compounds, I do in Berlin, and also I'm starting a collaboration with someone at FHML, Martin Honig. And finally, the bimetallic chemistry that I started was kind of done in collaboration with Marika Raya, who was a former colleague, and we kind of collaborated a bit on that project, and that was very nice indeed. And I'm very sorry that she left Maastricht, but we had a fruitful collaboration and published a paper together. And I've just started a collaboration with Dr. Akhilesh Verma at Cotton University in India, and he's doing a lot of work currently on this osmium chemistry. I presented some of those results tonight, but that's actually unpublished. I hope to be able to publish those results next year. I think that's going to be very nice. And of course, finally, a lot of students that I've had the privilege of working with. And yes, you know, this is, I think, very nice for them too, to actually do this research. You see, this has actually only been a 4-year period. I only started this chemistry 4 years ago, and I think we've made quite a lot of progress in a very short period of time. And so I think I'm looking forward actually to continuing this chemistry in the future, and particularly elucidating the biological activity and mechanisms of these compounds. And lastly, I'd actually like to thank the University of Maastricht and the Faculty of Science and Engineering, along with the Maastricht Science Programme for funding, and also SWOL, the University Fund of Limburg, which has made a small grant making some of this work possible. So I'm sorry for going a little bit over time, but I think that's a very quick and dirty, let's say, summary of what we've been doing. And yes, I think there's substantial progress, and I'm looking forward to more scientific progress in the future.
thank you, Burgert Blom, for this interesting talk. Again, for me, it's like I'm flapping my ears. What is he talking about? Well, I really love this idea of a playground where you try out little things. It's sometimes I really do like jigsaw puzzles, so I can imagine how you work and you try to connect certain compounds and to get this each better.
Rationality that goes into that, and I think that's important.
I think a lot of people think, we as chemists just mix things together and hope for the best. It's not true. We really rationally design things thinking that they're going to have certain biological functions. So yes, it's a process of design and then building. It's kind of like an architect. Well, yes.
I like that idea. It makes research such fun. Any questions from the audience? Yes.
Okay, that was really enjoyable. A lot of the more serious questions I had were answered like minutes later, so that was great. So I'm only kind of left with a few small questions on what we talked about. Some of them were really small, like you mentioned at some point that one of the cell lines was cisplatin resistant, and I was wondering what that was about.
Oh, that's a good question. Sometimes. Oh yes. The question was, some cell lines are cisplatin resistant. Why is that? Well, some types of cancer just happen to have developed a resistance to cisplatin. And actually, this is quite a big problem because there are certain cancers that are simply resistant to that chemical agent, and that cannot be used to treat them. So you have this notion of acquired resistance to particular drugs, cisplatin being one of them. And so that's why it's important that one develops other drugs, particularly for those types of cancer.
And then following on from that, another question was how you choose the cell lines, and I'm presuming that this resistance is part of that decision.
That's a very good question. I mean, as you can see, sorry. Yes, I'm sorry. The question was, how do we choose the respective cell lines? Well, so I have many collaborators, and each of them have specific interests in specific cell lines for particular reasons. For example, at the University of Cape Town, my colleagues are working particularly on breast cancer, okay, because it so happens that locally there's a very big problem with breast cancer. And so that's why they choose that specific cell line. My colleague in Switzerland usually uses ovarian cancer. So it really depends on who I'm actually collaborating with. And ideally what you'd really want is to test them on many different cell lines, but that's very costly. And so to gain insights into the biological activity, one typically selects a few cell lines and does a screening. And that gives you some idea of the biological activity. But indeed, on different cell lines, you have different activities.
And then you're choosing the metals like ruthenium and osmium. How do you choose those metals knowing what you want them to do? Is it entirely we'll try it, see something happens, and then try to determine the mechanism afterwards?
That's a very good question. It comes kind of what I said in the beginning. I'll repeat the question. How do we choose osmium or ruthenium? And is it a matter of trial and error? Well, yes and no. We know that ruthenium does have, that certain classes of ruthenium compounds do exhibit promising cytotoxic activity or anti-cancer activity. And so we use that as a basis to then further design molecules that might, according to our design plans, possibly have better activity. Now, for example, one of the things that's important is you would have seen all of these ruthenium compounds have this ring of carbon atoms sitting on it, kind of like a hat. That's actually like a benzene ring. And that's very important because it's thought that that ring is actually responsible for uptake in the cell through the membrane. It's fat soluble. It's a lipophilic substituent. And so it's important to design a specific substituent that's fat soluble that will enable the drug to actually be taken up in the cell. That's one of the considerations. And the other one, as you saw from my lecture, is you need to kind of balance, and that's what we've discovered, water solubility against fat solubility. So you need those lipophilic substituents for uptake, but at the same time, it would seem, based on our research, that you also need a certain degree of water solubility. And that seems to be a difficult balancing act. That's precisely why we moved in the direction of those germanate and stannate complexes, because they're ionic, but they still contain germanium and tin. So you still kind of have that possible effect and synergy from tin, which is also cytotoxic in its own right, but it's ionic, and you've got the lipophilic groups to ensure the uptake in the cell. So there is a degree of design that goes into it, and then when you test it, you see how your, let's say, design plans held out against the actual biological data. In the case of the iron ruthenium system, you saw that very nicely, that the idea there to attach iron to it to enhance uptake in the cell seems to have worked, because that's absolutely in line with what we saw with the biological activity. So there is design and rationality that goes into it, absolutely.
I just have two small questions left. So you mentioned lengthening the tether between the bimetallic compounds. What was your expectation for increasing the tether length?
Well, actually, that's a good question. We didn't really know. I was just interested to see if there is a... sorry, I keep forgetting. What was the rationale between increasing the length of the chain, and what was I expecting? The answer is, I just wanted to see if there's some relationship between the increase of the chain length and the cytotoxic activity, right? And I didn't really know what to expect, because it might be that you kind of have a, I don't know, some sort of a parabolic function where you have sort of a chain length that's optimal, and if it gets too long, then you again have a decrease in biological function. That's actually what I anticipated. Now, at this stage, we only kind of have 2 data points, so we can't really say either way which direction the trend goes. It would seem that going from 3 carbon atoms between the 2 metals to 4 decreases the cytotoxic activity slightly. But currently in our lab, we're actually making, hopefully, systems that have 4, 5, and 6 atoms between them, between iron and ruthenium. So what you saw in the case of the di-iron system, but we can't really compare those two because the one is iron and ruthenium and the other one is iron and iron, is there we had a particularly long chain, and there is a complete reduction in activity. But I suspect that's because ruthenium is not present, okay? So the short answer is we don't know. And essentially, we simply need more data to actually see what the structure-activity relationship is. And I do have students working on this currently, and they're in the audience.
And then my last question was, you mentioned the long-term stability of the compounds. Was that long-term stability like in vivo within the...
In vitro.
In vitro? Or was
It-
that like a shelf-life, shelf-life kind of...
So the question was, the in vitro long-term stability, is that sort of a measure of the shelf life? No. It's a measure of the stability. It's an experiment you can do to gain insights into how stable the drug is. It's kind of simulating how stable the drug is and for how long that drug operates once it's in a biological medium. It's not in vivo, it's in vitro, but it gives you some sort of an indication. And so it's not the shelf life of the drug, right? The shelf life would be the compound isolated in a bottle. These materials are very stable. We've checked that for months. But that experiment is looking specifically at the stability of the agent once it is in a biological system.
Any? Maybe from people? No? Okay, Burgert, thank you very much.
Thanks.
We wrap it up. Well, again, I didn't understand it directly, but I could follow your research, and I think it's very interesting. Also, the international cooperation.
That's very important.
and the work with students. So thank you for talking to us about this and the audience at home. Thank you for coming. I hope to see you next time at another lecture.
Thank you.